Okay, this is going to be the screencast about tour of the cell, numero dos. So let me share my screen. iPad power, let's go. Oh no. Let's go. All right. Whew. For all of you that listen to this at 1.5 speed, just kidding. All right, so we are going on. We talked last time about the genetic information of prokaryotic cells with this circular DNA versus eukaryotic cells with these single strands of DNA that are linear. We talked about prokaryotic versus eukaryotic cells. And now we are going into the eukaryotic nucleus. So again, for eukaryotic cells, they are larger than prokaryotic cells. They have linear DNA in a nucleus, and they have membrane-bound organelles. Organelles. And my face is hiding them. Membrane-bound organelles. I wonder. Anyway, all right, so now we're going to be talking about some of those membrane-bound organelles that occur in these more complex eukaryotic cells, right? So the eukaryotic nucleus, when you think nucleus, you should think that's where the DNA is, all right? And the nucleus is pretty special. It has the nuclear membrane, sometimes called the nuclear envelope, has two lipid bilayers. All right, two of them. So a normal lipid bilayer and then another lipid bilayer, all right? The outer one is continuous with the endoplasmic reticulum. And you can see that down here in this image here, how you can see that the outer nucleus is actually continuous with this other membrane structure called the endoplasmic reticulum. The nucleus also has these special kind of holes in them, and those are called the nuclear pores, all right? And the nuclear pores help get things inside and outside of the nucleus. Currently, the only thing we've really talked about is that we have DNA on the inside, right? DNA can be transcribed into RNA in the nucleus. That RNA can actually come out of the nuclear pores, right? And that RNA is going to be then important in making protein, all right? Okay, so we have the nuclear envelope with its two lipid bilayers, outside continuous with the ER. We have the nuclear pores, which help get things in and out of the nucleus. And we also have this thing called the nuclear lamina, which basically helps maintain the shape of the nucleus, all right? And this is composed of protein. So if you look here, this picture labeled A here is a normal nucleus by light microscopy, and then by scanning electron microscopy. And you can see those little nuclear pores or the holes. And what you can see on the background is this kind of matrix network. That's all the nuclear lamina. In somebody that has some genetic disorders, you have a nuclear lamina that isn't very strong, all right? And what happens, as you can see in this green image, is that the nucleus kind of can fold in on itself. And if you look in at the more high-powered microscope, you can see that, yes, you can still see nuclear pores, but you can also see all this black region, which means that the nuclear lamina is not really tight, all right? And that can lead to a really devastating genetic disease called progeria. This guy, who is so freaking awesome, his name is Sam Burns. He had progeria. He was a teenager. He spoke really eloquently about this disease. All right, ribosomes. Ribosomes really are protein factories, all right? So ribosomes, what they're gonna do is convert RNA into protein, right? Ribosomes are the sites for protein synthesis. They look a little bit like those little Hawaiian buns, those delicious little Hawaiian buns that can, like, they have the big top and then the bottom top. or the bottom, whatever. And these are made of both protein and ribosomal RNA, right? And remember that ribosomal RNA, this rRNA, is made in the nucleolus, which is at the very center. It's kind of like just a region of the eukaryotic nucleus, all right? So ribosomes made of protein and rRNA, and they can occur in two places in the cell. You can have free ribosomes, here that I'm boxing, that just are in the cytoplasm, or the cytosol, and you can have ribosomes that associate with the endoplasmic reticulum, giving you what is called a rough ER, all right? So ribosomes come in these two flavors: free ribosomes, rough, or the on the endoplasmic reticulum. So looking more at the endoplasmic reticulum, this is a part of the endomembrane system. So remember, the endomembrane system includes the nucleus, the ER, the Golgi, vacuoles, vesicles, and lysosomes. Lysosomes. So this is all the kind of membrane that can move throughout a cell, membrane compartments. So the endoplasmic reticulum, otherwise known as the ER, is part of the endomembrane system, and in fact it accounts for more than half of the total membrane in most cells. So it's big, and it's a lot of folded membrane. And it's, again, continuous with the outer membrane of the nuclear envelope, or the nuclear membrane, right? Just like ribosomes that come in two flavors, the ER also comes in two flavors. It can be on the smooth. It can be smooth, meaning it has no ribosomes. So actually I'll put that. So if it's a smooth ER, it has no ribose bound to it, right? That's involved in lipid synthesis. You can also have a rough ER where you have plus ribose bound to it, all right? Decorated with ribosomes, and that's really important, again, for protein synthesis. So the rough ER is important in making proteins, specifically proteins that we're gonna find out in the plasma membrane. So any protein, so here I'm drawing a little lipid bilayer. Let's say this is the plasma membrane at the outside of the cell. You can have proteins that are embedded in that, and those proteins come from the rough ER, right? So they're made in the rough ER. And they help transport vesicles throughout the cell, right? And we're gonna draw this out in class. But in general, there's kind of a flow of membrane from the ER, from the ER to vesicles, just little sacs of membrane, to the Golgi, out the Golgi to vesicles, and then out to the plasma membrane. And that together kind of is a lot of the endomembrane system. Okay, so the Golgi apparatus is, I always think of it as the post office, right? So if you are sending a letter, you put a zip code on it, you give it to the post office, and the post office does something magical and tells your letter to go to the right place. The Golgi does the same thing, all right? So it modifies lipids and proteins that are made from the ER, made on the ER. Again, they are proteins made on ribosomes, right? And again, those can decorate the ER to make the rough ER. And the Golgi makes some macromolecules, but in particular, sugars, right? It makes sugars, right? So what the Golgi does is, just like you put a zip code on things, the Golgi is going to add sugars to proteins to tell them where to go in the cell. So it sorts and packages materials into these little sacs or membrane sacs, for example, down here, which is a vesicle. It's just a phospholipid bilayer sac that might have like proteins in it or something. And it tags them with these sugars to send them to their final destination. And if you look at an actual Golgi, they have these long kind of folds. Those are called cisternae. And the Golgi has a cis side where things receive and a trans side where vesicles can bud off of and go to their particular place. Lysosomes, this is one that students usually really like. Lysosomes, I think, are the stomach of a cell, right? The stomach. So that means that they have, again, a membrane sac. So it is a phospholipid bilayer compartment within the cytoplasm that contains these digestive enzymes, all right? And these digestive enzymes basically can break down macromolecules, or they can eat them, all right? The lysosomal enzymes work best with a low pH, so meaning high acidity, meaning high amounts of hydrogens, very much like our stomachs. Our stomachs break down our food in an acidic environment. Lysosomes do the same. So if we look down at this picture, this is a process called phagocytosis. My students all like giggle when they hear that, but whatever. So phagocytosis is where a cell will take up food. So like a way of eating, the membrane bends in and then it'll form what's called a food vacuole. The lysosome, the stomach then, will bind to that food vacuole and you will get those enzymes working to break down that food, right? If a cell is starving, they will go through what is called autophagy or autophagy, where they will actually start digesting organelles within the cell. So cells can also starve, and they break down those food or themselves if they're starving through lysosomal degradation, right? So vacuoles are more just sacs. We just talked a second ago about this here, the food vacuole, which is a membrane, and it binds, or sorry, not binds, but a membrane that kind of holds cellular food, right? And a vacuole is just like a sac that's going to do that. However, vacuoles also come in many flavors, depending on what kind of cell you are. So you can have food vacuoles that are taken up by the cell through phagocytosis, right? The ability to take up material from the environment. You can also have what are called contractile vacuoles, and this is found in many different freshwater single-celled organisms. So here down here is a paramecium, and the paramecium, this little guy here, has a contractile vacuole that can pump out excess water. So it kind of contracts, and then it squirts water out. And I can show you that movie in class. And then there are central vacuoles, which are found inside plant cells, and they hold mostly water, but also some compounds. So in plants, let's just look down at this one first. If you have a plant that is very happy and turgid, meaning it's had a lot of water, then the central vacuole is very full, right? When you have plants that have not been watered very much, then that vacuole shrinks to something very small and you get a sad looking plant. And that's called flaccid. And that's when the plant is not very happy, doesn't have enough water. All right, now we come to our two big, bad, awesome organelles, which are the energy converters. And we're gonna be talking about these a lot. So you're gonna get to know mitochondria and chloroplasts very, very well. Let's start with mitochondria. So first off, mitochondria is in every single eukaryotic cell, all of them. It's a requirement. It is double membraned, and you can see that down here in the image. You have so you have an outer membrane, and then you have an inner membrane, right? Outer and an inner membranes. this is, the mitochondria represent sites of cellular respiration. And what I mean by that is that humans breathe in oxygen, and we can then break down glucose, C6H12O6, and we can make energy in the form of ATP, all right? So mitochondria are really important in both plant and animal, all eukaryotic cells, in creating energy by breaking down glucose using oxygen. And that happens in our mitochondria. Chloroplasts, on the other hand, only occur in plants. So these are in plants and algae. You also see a double membrane. So you can see an outer membrane down here. So you have an outer, and then you have kind of stacks. Actually, here. Sorry, rewind. You have an outer membrane, you have an inner membrane, and then you also have these kind of weird stacks on the inside, which we'll talk more about when we get to photosynthesis. But they're double membraned. They contain a green pigment called chlorophyll. And these are the sites of photosynthesis. This is where plants use light energy and carbon dioxide to create glucose and oxygen. How awesome for us, because we then use that oxygen and glucose to make ATP. All right, so mitochondria and chloroplasts have many, many similarities with bacteria. And when I mean bacteria, I mean prokaryotic cells, all right? Here are the similarities, they have many similarities. Some of those is that they contain free ribosomes, right? And they have circular DNA. Mitochondria and chloroplasts, the organelles within a cell, they can grow and reproduce independently within a cell. And something interesting about them is that they're double membraned. That's unique for organelles. So it turns out, and we're going to talk more about this in class, and the Amoeba sisters have an awesome little video that I've posted about this as well. Lynn Margulis, a great scientist, came up with this idea called the endosymbiont theory, which states that early eukaryotic cells, right, engulfed a prokaryotic cell, which then formed a kind of symbiotic relationship, eventually becoming its own organelle. So let me explain that again, and then don't worry, we're going to talk about this in class. But here we have kind of one of the very first eukaryotic cells. So we'll call this the first eukaryotic cell. And that eukaryotic cell actually ate or phagocytosed a prokaryotic cell. It did not eat a mitochondria, right? It ate a prokaryotic cell, which then the cell decided to keep that thing around. Instead of digesting it, they're like, wait a minute, this mitochondria, or this prokaryotic cell can give me energy. So I'm just going to keep that around as an organelle. Then later on, that cell ate another prokaryotic cell. But this prokaryotic cell had the ability to photosynthesize. It had green pigment that allowed it to photosynthesize. And so that cell was like, hold on, I have a mitochondria that can give me energy. That's pretty sweet, because that first prokaryotic cell evolved into this little mitochondria. Now, this little prokaryotic cell that I just ate, I'm going to keep that around too, because it can do something cool for me. In other words, photosynthesize. So that's the endosymbiont theory. There's a great movie on that. We can talk about that more in class. We can even draw the whole thing out if you want. Okay, then we have peroxisomes. Peroxisomes are my least favorite organelle. Don't tell people that love peroxisomes. I don't really care about them. They're specialized compartments. They have a single membrane around them. They are for the breakdown of fatty acids, and they can produce hydrogen peroxide and make it into water with a specific enzyme. So they have an enzyme in there that can break down hydrogen peroxide and convert it to water. In one of the labs, our enzyme lab, we kind of use one of these enzymes called catalase, which does just this, all right? And that can be found in peroxisomes. Other than that, All right, the cytoskeleton. This is one of my personal favorites. What it is, it's a network of fibers that that are throughout the cell. And these guys provide mechanical support and help maintain the shape of a cell. So for example, if we have a cell with its nucleus, and we have its ER, and we have its Golgi, and we have some mitochondria, we have some lysosomes, we have some vacuoles, etc. We have some vesicles. But that cell has a cytoplasm that's just like an aqueous environment, kind of jello-like actually. That doesn't have a lot of shape to it. So what we have is we have these blue things here, which are microtubules. They're here. They're large, they're hollow tubes. They are really important in transporting things around the cell. Then we have actin. The actin is around the edges of a cell, kind of keeping that shape. It's also throughout the cell. That is called actin, sometimes called microfilaments. These are long, thin, solid ropes for the structure. Those are really important in muscles, actually. And then we have our intermediate filaments, this third one, and those are usually right at the rim of a cell as well. And they're called intermediate filaments because they're intermediate size between microtubules and actin. And the one thing that you should know about the intermediate filament is that that's the nuclear lamina that helps determine the shape of the nucleus. The nuclear lamina is our intermediate filaments, right? One thing that I failed to tell people last year, the cytoskeleton, these are all proteins, right? The cytoskeleton, these are all proteins that help maintain the shape and support for a cell. Okay, so when we look at the cell, and I kind of drew, so we're gonna be, sorry. We are looking here, and again, these are in only animal cells. We are going to talk about centrosomes and centrioles. And this is what I did my entire PhD on, are studying these little buggers, which are the centrioles. All right, so a centrosome is a microtubule organizing center. All right, so what you can see down here is the little black part that I'm drawing. That is going to be the centrosome. The centrosome is kind of this amorphous thing. Inside of it, you have two centrioles. Together, they are responsible for growing microtubules, right? Those long, hollow tubes. And that is really important for the structure of the cell. So here in A, you see that the microtubules are important for structure for that cell. When you have a dividing cell, we're going to see that microtubules are really important in separating cells when they divide. And microtubules are also really important for the making of cilia and flagella, which we'll talk about in a second. But wherever you have microtubules growing out, you have a centrosome or centrioles, which are at the heart of the centrosome. So cilia and flagella. So cilia and flagella really represent, when I put this, an extension of the plasma membrane. So long, thin extensions of the plasma membrane. Don't worry about this. I don't care about that you know about basal bodies. A long, thin extension of the plasma membrane. And these are important for movement or cell motility. And you have some experience with this. So sperm, you guys have all heard about sperm cells, right? Where you have a single cell with this long whip-like thing in the back. That is a flagella. If you zoom in on that, that is those are microtubules around a plasma membrane, right? So microtubules around a plasma membrane, and they can form those whips. And I can show you this video in class. And in the paramecium, which again has that central vacuole necessary to kind of like expel water. But if you look on the ends, and I'm just kind of drawing a more macro view of them, these are cilia. Flagella, cilia, exactly the same thing. Flagella, you have one or two. Cilia, you have thousands, right? Cilia are really important in our fallopian tubes to move those little eggs along. In our throats, when we inhale and we get something kind of stuck in our throats, the cilia helps deal with that. All right, then we have the extracellular components, all right? These are secreted by the cell, right? Cells make things and they secrete them out. And this is really important for support, adhesion, don't worry about cellular motion. And the big examples of this are the cell wall, which how awesome that you know a little bit about a cell wall already. It's made of cellulose. It actually occurs on the outside of the plasma membrane. So if we have the plasma membrane of a plant cell there, then we have this rigid cell wall outside. So what that means is that it was secreted out of the cell in order to make it, right? The ECM is the extracellular matrix. And this is a lot of proteins and polysaccharides that kind of exist on the outside of a cell wall or outside of a cell. So if you have a cell, there are kind of fibers on the outside that will help, for example, bring things in and out. They will help other cells communicate with them. That's the ECM. And then we have junctions, which if we have two cells that need to communicate with each other, they form kind of adhesion with each other. And those are the cellular junctions, right? And again, all of these are secreted out of the cell. So they're extracellular on the outside of the plasma membrane. All right, and we will stop there.